Okay, good morning everyone. Good morning, good morning. Good morning all of you. Good morning, good morning. Okay. If anyone misses yesterday classes, please watch it especially, okay? Especially the class where we have discussed about the carbohydrate metabolism regulation, and I want especially you people to watch the differences between the hexokinase enzyme as well as the glucokinase enzyme, what we have studied with respect to the graph. Okay, I want you people to watch that without fail. Okay, so now in the yesterday class, friends, we have what we completed in the yesterday class. In the yesterday class, we have completed the glycogenesis, we have completed the glycogenolysis. What we completed, glycogenolysis, and most importantly we have completed the PPP pathway, and we completed the regulation of carbohydrate metabolism, right? So till here we have completed in the yesterday class, is it? Yes. So friends, now let's try to understand another most important concept, that is the TCA cycle. This time whatever the question we got in the September 2022 paper, it's a very straightforward question, and that too matching question, and very very easy question also they have asked. But friends, every time we don't get that much easy type of question, but there is a chance, there is a possibility we can get the question in a very direct way also. So all the students, listen to me. What we have to read in the TCA cycle, first we have to read the location, means all where this particular reactions are taking place. Then second step, we have to read all the enzymes involved in the TCA cycle. And most important, we have to read the key regulation process. And very important, where we got the question is, that is about the amphibolic pathway. Okay, so these are all the four concepts, friends, we have to read very briefly and elaboratively. Okay, let's start. TCA cycle. So everyone please look into this. Actually, what will be the fate of this particular pyruvate, friends? If everyone of you quickly recall, once we produce the pyruvate, what is happening to the pyruvate? Pyruvate only in the presence of oxygen. What, friends? Pyruvate only in the presence of oxygen, it is converting into what, friends? Acetyl CoA. That is what we understood. Pyruvate in the absence of oxygen, in case of a muscles, it is converting into lactate. And pyruvate in the yeast, it is getting converted into what? Ethanol. Now we discussed whether it is a, understand my words correctly, friends, whether it is a carbohydrates, whether it is a amino acid, whether it is a fatty acid, everything, all their fate exclusively depending on what, friends? Acetyl CoA, is it? That is the reason, friends, what do we call, you can call the acetyl CoA is a key intermediate molecule. What, friends? Acetyl CoA is a key intermediate molecule that is involved in this process. Okay, please don't get confused with that particular point. Okay. Now, where this particular Krebs cycle is taking place, friends, of course it is taking place in the mitochondria. We can call it as the citric acid cycle, we can call it as the TCA cycle, or the Krebs cycle, based on the name of the Adolf Krebs, which he awarded Nobel Prize for this. Friends, even you can expect the question from here also. Okay, I want every one of you to remember compulsory, friends, what are the major key events that are taking place within the mitochondria. I mean to say at different locations of the mitochondria. I want every one of you to remember this. We know that mitochondria is a double membrane organelle having the outer membrane, having the inner membrane, and friends, the space between the outer and inner membrane, we will be calling it as the intermembrane space. Is it, friends? And most importantly, the fluid filled structure. Everyone understand, friends? The fluid filled structure, you can also call it as a gel like structure, or our friends, cytoplasm like structure, which is present inside, what do we call it as generally, matrix. Why why we have to study different type of structures? The reason is very simple, friends, because different reactions are taking place at different parts. That's the reason we have to study everything. Okay, so the first point, everyone remember my words. The first point every one of you has to remember, the moment you heard the name of a outer membrane, just keep it in your mind, friends. Outer membrane is the freely permeable membrane. Outer membrane is what, friends? Outer membrane is the freely permeable membrane. Why do we call it as the freely permeable membrane? Because it is allowing almost all the ions as well as almost all the molecules to cross this particular membrane. That's why, friends, we can call outer membrane is the freely permeable membrane. Do not get confused with that particular point. Then, friends, what about the inner membrane? All the students listen my words correctly. I never use the word inner membrane is permeable to all the ions and all the molecules like outer membrane. No. You have to remember my words that inner membrane, friends, highly impermeable. Friends, highly impermeable in such a way that, friends, even it never allow small molecules also, it never allow small ions also, and friends, most importantly, very importantly, even the inner membrane of mitochondria never ever allow the H plus ions also. This is a very important point, friends. Outer membrane allows everything, friends, but the inner membrane is not allowing anything, including the small molecules, small ions, as well as the H plus ions also. And you know, no, friends, where does the electron transport chain will takes place, friends? We all people studied very clearly. ETC, electron transport chain, is taking place in the inner membrane of the mitochondria only. What is the reason? What is the reason electron transport chain is taking place in the inner membrane of mitochondria? The reason is very simple. Several electron acceptor complexes 1 to 4. Several electron acceptor complexes 1 to 4. We know that we are going to study in the electron transport chain, there are 4 electron acceptor complexes are present. All those 4 complexes are present on the inner membrane of mitochondria only. That is the reason ETC will take place over there. And the most important enzymatic complex, which we can call it as ATP synthase enzyme. We people widely call it as what, friends? F0 F1 complex, which is involved in the production of ATP molecule. Where it is present, friends? Of course, it is also present in the inner membrane of the mitochondria only. Please don't get confused with the statement. Okay, now listen, friends. Matrix. Matrix is the region where most of biochemical reactions will occur. Matrix is the region where most of biochemical reactions are taking place. Friends, please do not confuse with this particular point. All the students follow my words correctly. If you quickly recall, I have told to all of you that, friends, where the pyruvate is going to be converted into acetyl-CoA, of course, friends, it is happening in the mitochondrial matrix only. Why? Because mitochondrial matrix is the region where the pyruvate dehydrogenase enzyme complex is present. And the second one, where the Krebs cycle is taking place, undoubtedly the Krebs cycle is taking place in the matrix only. What will be the reason? The reason is very simple. Whatever the enzymes that are required for the Krebs cycle, all the enzymes are present in the mitochondrial matrix only. Friends, I want you to remember, what is glycolysis, friends? Glycolysis is a process where the oxidation of glucose is happening. That is, breakdown of glucose is happening. Similarly, friends, what do you called breakdown of the fatty acids? Breakdown of the fatty acids, we generally call it as, friends, beta oxidation. Now once again, the answer is very clear. Why the beta oxidation is taking place only here? What will be the reason? Obviously the reason is that all the enzymes required for the beta oxidation also present in the mitochondrial matrix only. There's no doubt about it. Friends, understand, not only Krebs cycle, not only fatty acid breakdown, and friends, even if you look into the amino acid oxidation, I want you people to remember everything, friends. Even amino acid breakdown is also happening in the mitochondrial matrix only. Please don't get confused with this particular point. And we all people know that mitochondria is said to be what organelle, friends? Mitochondria said to be self-replicating organelle. Correct now, friends? Which means mitochondria having its own DNA, mitochondria having its own ribosomes. The question is very clear, where this particular DNA is present. Where this particular ribosomes are present. Obviously, friends, they are present in the matrix only. Don't get confused with that. I want you to remember every point whichever I am writing here. Apart from this, friends, certain cofactors such as ATP, ADP, magnesium, calcium, all these are all present in the mitochondrial matrix only. Not only this, friends, a very important point, several apoptotic enzymes. Several apoptotic enzymes, especially the enzymes called as caspases. Especially the enzymes called as caspases, they are exclusively present in this particular matrix only. Friends, please don't get confused with that. So I'll quickly summarize what are present in the matrix. Friends, try to understand, matrix is the region where most of the biochemical reactions are taking place, where the pyruvate converted into acetyl-CoA because of the PDH, Krebs cycle occur, beta oxidation, amino acid breakdown, and apoptotic event, that is cell death events, occur because of the presence of a caspases enzymes. And since the DNA is a self-replicating organelle, DNA, ribosome, ATP, ADP, PI, magnesium, calcium, potassium, all will be present in the mitochondrial matrix only. So that is the reason matrix is the area most of the biochemical reactions are exclusively taking place. Okay. Now let's try to understand the Krebs cycle very briefly and elaboratively. Okay. So friends, this is just an overview. This is just an overview of the cycle, and once again we will try to study every single step. First, let's study the overview. Now every one of you tell me, pyruvate. How many pyruvates we produced in the glycolysis, friends? We all people know that we are producing 2 pyruvates in the Krebs cycle, is it? So friends, from the 2 pyruvate molecules, how many acetyl-CoA we are going to produce, friends? We all people know that from the 2 pyruvate molecules, friends, 2 acetyl-CoAs are releasing. So most of the students, what they will do, they will only calculate for 1 acetyl-CoA all the time. No, you should always calculate for the 2 acetyl-CoA. While considering, you consider for 1, But while doing the process of calculation, you should always calculate for the 2-acetyl-CoA. But while considering initially, not to get confused, you can consider for 1-acetyl-CoA. No problem in that. Understand my words. So friends, acetyl-CoA is how many carbon containing compound? Acetyl-CoA is a 2 carbon containing compound. Friends, all the students listen. I am considering 1, but while I am calculating, friends, undoubtedly I will calculate for the 2. Okay. This is a just overview. Listen to me carefully. Oxaloacetic acid combined with the acetyl-CoA. Acetyl-CoA is how many carbon, friends? 2 carbon. And you all people know that oxaloacetic acid is how many carbon? It is easy to remember. And every student has a Krebs cycle without fail. Because you can expect the question even from the structure also. Many times asked question. Oxaloacetic acid, which is a 4 carbon, combined with the acetyl-CoA, which is the 2 carbon. So 4 plus 2 total will become what, friends? 6 carbon. All the students remember. What is that 6 carbon containing compound? Citrate. Understand, friends, very correctly. So the stable product or understand, friends, the stable product or the intermediate product produced is how many carbon containing compound? 6 carbon containing compound. What is that? Citrate. That is the reason we have called it as citric acid cycle, because the intermediate produced is the 6 carbon. That is the reason only we are calling it as the citric acid cycle. Don't get confused, friends. So citrate is a how many carbon containing compound, friends? 6 carbon containing compound. So what is happening, friends? The citrate can be converted into its isomer. Friends, don't worry, I will be explaining every step elaboratively, but try to understand this overview. Citrate can be converted into its isomer. We can call it isocitrate. Friends, isocitrate is how many carbon? Undoubtedly, isocitrate is also a 6 carbon, right? There is no change in the carbon number. What has happened? Just citrate converted into its isomeric form, which we called isocitrate. That's all. Now, the isocitrate, from here every student follow me, I'll explain. Isocitrate, which is a 6 carbon containing compound, friends, it is converting into 5 carbon containing compound. So what is that 5 carbon containing compound, friends? We know that alpha ketoglutarate. Now isocitrate is a 6 carbon, alpha ketoglutarate is a 5 carbon. Then how many carbons are left out, friends? There is a release of only 1 carbon. Yes or no? Now all the students remember here. Understand my words correctly. Whenever isocitrate can be converting into alpha ketoglutarate, friends, NAD plus reduced to the NADH. Remember every student. So there will be a production of the first NADH here. Now next one, alpha ketoglutarate can be converted into what, friends? Succinyl-CoA. Tell me, friends, alpha ketoglutarate is how many carbon we studied? 5 carbon. And friends, succinyl-CoA is a how many carbon, friends? 4 carbon containing compound. Which means, friends, there is a presence of one more carbon dioxide. So that carbon dioxide, that carbon can be released in the form of a carbon dioxide. Remember, friends, in this step, there is a production of the second NADH molecule. Don't confuse. Now look into this point very correctly, friends. Succinyl-CoA can be converted into what? Understand the concept. Succinyl-CoA can be converted into succinate. Now tell me, friends, succinate is a how many carbon containing compound? We all people know that, friends, succinate is the 4 carbon containing compound. Yes or no? Of course, yes. Succinyl-CoA converted into succinate. Succinate is a what, friends? A 4 carbon containing compound. Now tell me what happened. Everyone understand my words. Just CoA got released. All the students follow my words. Just CoA got released. And you know very well, the CoA is a energy rich precursor. What friends? The CoA is the energy rich precursor. Okay, the question is very clear. From this particular CoA, who is gaining the energy? That is the point. Friends, from the CoA, the energy can be gained by the GDP. So GDP combined with PI, friends, then there will be a formation of the ZTP molecule. Friends, don't get confused with this particular point. Understand once again, when the succinyl CoA converted into succinate, there is a no change in the carbon number. Since the CoA is releasing, CoA is an energy rich precursor. That energy can be utilized by the GDP and PI to form this particular GTP molecule. That's the point I want you to remember. Now what is happening in the next step? Succinate can be converted into what, friends? Succinate can be converted into the fumarate. Friends, succinate is how many carbon? 4 carbon. Fumarate is how many carbon? 4 carbon. There is no change in the carbon number. Just reduction is happening. What is happening? Just reduction is happening. And friends, here please do not get confused, friends. Here the reducing equivalent produced is not NADH. The reducing equivalent produced is what, friends? FADH2. Don't get confused with this particular point. And friends, what is happening to this particular fumarate? Fumarate is converted into malate. Malate is how many carbon, friends? Of course, 4 carbon. And finally, malate get reduced to what, friends? Oxaloacetic acid. That is also how many carbon? 4 carbon containing compound only. Whenever malate is reduced to the oxaloacetic acid, once again there is a production of the NADH, which is a third NADH. So friends, wherever we started the cycle, we came to the same region. That is the reason only we are calling it as the cycle. Now all the students look into this. How many, see friends, remember, right, I am considering for 1-acetyl-CoA. I am considering for 1-acetyl-CoA, but while doing the process of calculation, I will always consider for the 2-acetyl-CoA. So for 1-acetyl-CoA in the cycle, how many NADH are produced? 1, 2 and 3 NADH are produced. Is it? How many FADH2s are produced, friends? There is only one FADH2 will be formed. How many GTP molecules are formed, friends? Yes, there is a formation of only one GTP molecule. Friends, do not get confused with this particular calculation at all. Now friends, we will be discussing every single structure. Why we have to study every single structure? Because friends, more than 3 to 4 times the direct question was appeared from the structure itself. So that is the reason I am requesting every student to remember the structure. Friends, it is easy to remember the structures also. It is not that much difficult also, friends. You only tell me, in the Krebs cycle, everyone, in the Krebs cycle, how many 6 carbon containing compounds are formed? Easy to remember. There are only 2 6 carbon containing compounds are formed. Yes or no? One is the citrate, one is the isocitrate. Both are what? 6 carbon containing compounds. Correct, na? Then next, alpha ketoglutarate. Remember my words correctly. In the entire Krebs cycle, follow me friends. In the entire Krebs cycle, the only 5 carbon containing compound is what? Only one. That is the alpha ketoglutarate. And it is very easy to remember. In the structure, wherever you come across with the CoA, that is the succinyl CoA. Then the only thing you have to remember is 3 structures, friends. Succinate, fumarate, malate, anyway oxaloacetate is the starting compound. So remember friends, 4 compounds are 4 carbon containing compound, 2 compounds are 6 carbon containing compound, 1 compound is a 5 carbon containing compound. Friends, please do not get confused with that particular point. And there is a 1 CoA molecule. Very easy to remember, but you have to remember compulsory. And most important for the exam point of view, enzymes. Every single enzyme that is present in the Krebs cycle, you people has to remember. Okay, I'll try to explain every single structure now elaboratively, friends. Please try to pay attention. I want you to remember these all the steps correctly. Oxaloacetate, how many carbon? 1, 2, 3, 4 carbon. Combining with what, friends? Combining with acetyl-CoA. How many carbon acetyl-CoA? 2 carbon. Giving rise to what, friends? Citryl-CoA. Very simple, right? What is happening? Tell me, friends. Addition of a acetyl group, where I am adding the acetyl group just to this particular keto double bond. As a result, what is happening, friends? I am going to get a citrate, a very simple, which is an intermediate compound. And what we discuss in next, this particular citrate is going to be converted into its isomer. What is that isomer called as? Isocitrate. Is it, friends? By an important enzyme which we can call aconitase. It is very easy, friends. All the students look into the structure carefully. From the citrate, what happened first? All of you pay attention, friends. From the citrate, what happened first? From the citrate, there is a loss of water molecule first. Upon losing the water molecule from the citrate, citrate converted into cis aconitate. Now once again, what I am doing, to the cis aconitate, once again I have added the water molecule. See here, upon adding the water molecule, once again it is produced to what? It is producing the isocitrate. Friends, it's a very easy to remember. First citrate lose the water molecule, converted into cis aconitate, which is an intermediate. Now this cis aconate, upon addition of the water molecule, again it forms the isocitrate. What's the major principal enzyme involved here? The enzyme involved is the aconitase. And friends, a very important enzyme now, isocitrate dehydrogenase. What is the reason I am calling it as the most important enzyme? The simple reason is what, friends? It is one of the enzyme which is performing the irreversible reaction. That is the reason I am calling it as the most important enzyme. All the students look into the diagram carefully, friends. Isocitrate is how many carbon containing compound we just studied? 1, 2, 3, 4, 5, 6 carbon containing compound. This isocitrate converted into what? Alpha ketoglutarate. Correct now, friends? Now all the students follow my words correctly. When the isocitrate converted into alpha ketoglutarate, see, look into the picture, every student. Understand, it is what reaction? Decarboxylation reaction. Correct na? Which means I am removing the carbon. Yes or no? Of course, yes. So whatever the carbon that we have removed, what is happening to that carbon? It is releasing in the form of a carbon dioxide. And friends, please remember my words. Here what is happening? Look into the picture. The NAD plus is accepting this H. What, friends? The NAD plus accepting this H, and it can form this particular NADH2. Don't get confused with that point. You know what is the one more hint every student has to remember for the exam? Wherever you heard the dehydrogenase, one common point has to remember that there is a production of the NADH2. Remember that particular point. And you tell me, friends, can we bring back alpha ketoglutarate into the isocitrate? Is that possible? No, we can't do this particular thing. So that is the reason only we are calling it as the irreversible reaction. And friends, please don't confuse one more important point. In the entire Krebs cycle, they can ask, this is also in a part B question, how many number of oxidation reduction reactions that has been taking place in the Krebs cycle. Every student remember, friends, In the Krebs cycle, there will be always 4 oxidation reduction reactions are taking place. Never ever confuse with this particular point, friends. Yeah, see, why 4 oxidation reduction reaction? In 3 reactions, 3 NADH molecules are producing, and in 1 reaction, 1 FADH2 molecule is producing. Correct, na? That is the reason, friends, we are calling it as the 4 oxidation reduction reactions. So, among that 4, the first oxidation reduction reaction is what, friends? Isocitrate dehydrogenase enzyme. Don't get confused with that particular point. So, tell me finally what we have done. We have converted isocitrate to alpha ketoglutarate, which is a 5 carbon containing compound. Now, friends, another very important one, which is the second oxidation reduction reaction. Which is the second oxidation reduction reaction, right? What is this reaction? All the students look into this. Alpha ketoglutarate, which is a 5 carbon containing compound, it is converting into what, friends? It is converting into the succinyl CoA. Succinyl CoA is how many carbon, friends? 4 carbon. Now you only tell me, friends, there is a 1 carbon is getting released. So what is happening to that particular 1 carbon, friends? That 1 carbon released in the form of carbon dioxide. Now once again, what is happening here? NAD is getting reduced to the NADH. Friends, don't confuse with my words. You know, when we are reading the pyruvate dehydrogenase enzyme, we will understand this. Remember my words, don't confuse. After the ETC, we will study. Pyruvate can be converted into acetyl CoA. By what enzyme complex? Pyruvate converted into acetyl CoA by an enzyme complex called as PDH. Pyruvate dehydrogenase complex. I told you last time, we study next. Pyruvate dehydrogenase complex having 3 enzymatic subunits. We can call it E1, E2, E3. Why I am talking pyruvate dehydrogenase complex is whatever the subunits that are present in the pyruvate dehydrogenase complex, friends, understand my words, the same or the similar type, follow me correctly, the similar type of the enzyme complexes are also present in the alpha ketoglutarate dehydrogenase. This is the point I want you people to remember. So same kind of a coenzymes will be present. What are those? Is there any necessity to remember this particular coenzymes? It is mandatory. Okay, it is compulsory. You people has to remember them. Remember, E1, E2, E3. Alpha ketoglutarate dehydrogenase, dihydrolipoyl succinyl transferase, dihydrolipoyl dehydrogenase. I really don't know what you do, friends. It's your responsibility to remember them compulsory. Okay, friends, the first subunit, alpha ketoglutarate dehydrogenase, if it wants to be active, compulsory or mandatorily, friends, it requires the cofactor TPP. Without the cofactor TPP, never ever be active. Second point, dihydrolipoyl succinyl transferase. If it wants to be active, compulsory, it requires the lipoamide as their prosthetic group. Prosthetic group means, you know, right, which is the non-protein group, nothing but a cofactor or coenzyme we are talking about. Dihydrolipoyl dehydrogenase, which is the third subunit, compulsory require the FAD. Friends, do not get confused. And this is what reaction? Second oxidoreduction reaction. Once again, we produced the NADH. Now I told you very clearly. All the students follow me. Succinyl CoA is a how many carbon containing compound? 4. And succinyl CoA giving rise to what, friends? It is giving rise to the succinate only. Succinate is how many carbon, friends? Succinate is also a 4 carbon containing compound only. But what happened? All the students look at the diagram carefully, friends. Every one of you. This is the CoA. CoA consists of a what bond, friends? That is important for the exam. CoA consists of thioester bond. CoA consists of the thioester bond. All the students remember, friends, thioester bond always store or always conserve free energy. Always conserve the free energy. See, please don't get confused, friends. In case of a, understand my words very correctly, friends. In most of the organism, the free energy will be conserved in the form of a GTP or ATP. Friends, don't confuse my words correctly. 90% of the cases, the free energy can be stored either in the GTP or in the ATP. In the higher animals. In the higher animals. But if you look into a bacteria, friends, you have to remember that this particular higher energy can be used only in the production of ATP, not the GTP. But in higher animals, 90% of the cases it can be used for the production of GTP. In 10% of the cases it will be used for the production of the ATP. Friends, please don't confuse with this particular statement at all. Remember, the thioester bond which store the free energy that is used by the GDP and PI, hence there is a formation of GTP and this CoA get released out. Nothing happened. Just CoA released out. When the CoA is released out, the energy is also released. So finally succinyl CoA converted into the succinate. Now friends, one more important. All the students has to follow me correctly. Where is the first oxidoreduction reaction occur? This is the first oxidoreduction reaction. Where is the second oxidoreduction reaction takes place? Follow me, friends. This is the second oxidoreduction reaction. Where is the third one? This is the third one. And friends, in this oxidoreduction reaction, we are not producing the reducing equivalent NADH, we are producing the reducing equivalent FADH2. All the students follow my words correctly. Friends, succinate is a how many carbon containing compound? We know that 4 carbon containing compound. And friends, fumarate is a how many carbon containing compound? Of course, fumarate is also a 4 carbon containing compound. But all the students listen my statements correctly. What is happening here? There is an enzyme called as succinate dehydrogenase enzyme. What it is taking, friends? It is taking the H ions. It is taking the H from the succinate, and that H can be taken by the FAD. FAD undergo the process of FAD reducer to FADH2. What is simply happening, friends? Here simply happening, dehydrogenation is happening. Yes or no, friends? What is happening? Dehydrogenation is happening, which means there is a removal of the hydrogen ion. Who is accepting that? FAD. Very simple, right? So electrons are transferred from succinate to FAD. And friends, in the electron transport chain also I remind to all of you, but I want you to remember here itself. Electron is passing from succinate to FAD, and from FAD to where the electron will transfer. We will read in the electron transport chain. Electron will transport to the ubiquinone. No, that is a function of an enzyme. That is the nature of enzyme. You know, no, Archana, what is the function of transferase enzymes? They are the one which are involved in the transfer of certain functional groups. What is the function of a hydrolase enzyme? They are involved in breaking certain bonds. Similarly, that is the nature of an enzyme. What is the function of a deaminase enzyme? Which remove the amine group. So when I use a word dehydrogenase enzyme, they are the enzyme which are removing the hydrogens, and that hydrogens can be accepted by the NADH and FAD. That is the reason we get the reducing equivalent. That's the nature of enzyme, common. Okay, I want you people to remember, friends, from the succinate electron will be transferred to FAD, and we will discuss in the electron transport chain. From the FAD, electron will be transported to, friends, ubiquinone. Please do not get confused with this particular point. Okay, now let's talk about it. Fumarase. All the students look into this. Fumarate is how many carbon, friends? 4 carbon. And malate is how many carbon? 4 carbon. What is happening? There is no change in the carbon number. What happened? Look into the picture, every one of you, very carefully. This is what? Double bond. To the double bond, just I have added the water molecule, nothing else. Upon adding the double bond to the water molecule, the fumarate is just converting into its isomer, which we generally call it as what, friends? Malate. Please don't get confused with this particular point. So, stereospecific trans addition of a water to the double bond of a fumarate to form the L-malate. That is what exactly the function of a fumarase. Now remember, this is the 4th oxidoreduction reaction. Now what is happening to the malate? All the students listen, same. Malate is converted into oxaloacetic acid. Malate is a 4 carbon. Oxaloacetic acid is also a 4 carbon. What is happening once again? There is a removal of this particular H+. So once again the enzyme involved here, what? Dehydrogenase enzyme. The dehydrogenase enzyme, from the malate it will take out the H, that is accepted by NAD+, and it is giving rise to what, friends? Oxaloacetate. Don't get confused with that. Where we started our cycle? We started our cycle at the oxaloacetate and we reached the oxaloacetate once again. So friends, now let's quickly look into the stoichiometry or the calculation. Okay. Now all of you remember how I calculate individually. Okay, you have to remember individually also. You have to remember overall also. Okay. So first let's consider different type of steps that are involved. Okay. What are the steps that we people has to calculate? We have to calculate. I'm telling you the differences. Please remember. We have to calculate for the glycolysis. We have to calculate for the Krebs cycle, and we have to calculate for the electron transport chain. Sorry. Before considering the Krebs cycle, we have to compulsory consider a reaction. Is it? Which we can call it as what, friends? Pyruvate decarboxylation reaction, which means where the pyruvate is converted into acetyl-CoA. Without acetyl-CoA, we can't start the Krebs cycle, right, friends? That's why we have to compulsory consider the pyruvate decarboxylation without fail. Okay. Now let's calculate all individually. Then we can add all. Okay. So first, what we will consider? First, we consider the direct ATP. Then we will consider direct NADH or FADH2. Okay, let's take ATP or GTP, and we consider NADH, and let's consider FADH2. Then let's take the total. Okay, you all people know that in the process of a glycolysis, how many net molecules of ATP we produced? I am always using the word net. 2 molecules of net ATP we produced. Is there any production of a net molecules of GTP, friends? No way. There's no production of net molecules of GTP. What about the NADH? We have studied, right? The 6th step of glycolysis. Which step, friends? The 6th step of glycolysis, we produce 2 NADH molecule. You all people know that 2 into 3, 6 molecules of ATP. Is there any production of FADH2 in the glycolysis, friends? There is no way. So 6 plus 2 total 8 ATP we got only in the glycolysis. Most of the students will will leave this pyruvate decarboxylation. Tell me friends, in the pyruvate decarboxylation, did we produce any ATP molecule directly? No. In the pyruvate decarboxylation, did we produce any GTP molecule directly or indirectly? No. And in the pyruvate decarboxylation, how many NADH we produced? You know, right? Pyruvate is a 3 carbon and pyruvate converted into 2 carbon acetyl-CoA. Then there is a production of 1 NADH, right? So since 2 pyruvates are converting into 2 acetyl-CoA, then there is a production of 2 NADH. So 2 into 3, 6 molecules of ATP. Is there any FADH2 producer, friends? No way. So total how many ATP we produced here? 6 ATP. In which reaction? Pyruvate decarboxylation reaction. So friends, what will be the next one? Krebs cycle. All the students follow me correctly. In the Krebs cycle, we did not get any ATP directly. Correct na? See friends, in the Krebs cycle, currently I am talking about 1 acetyl-CoA. We will directly multiply at the last for the 2 acetyl-CoA. Okay. To avoid the confusion, let's calculate initially for 1 acetyl-CoA molecule only. Okay. So we do not produce any direct ATP molecule, but we know that we produced 1 GTP molecule. Anyway, friends, we all people know that 1 GTP molecule is ultimately giving rise to the 1 ATP molecule only. We know that. I am talking about 1 acetyl-CoA. So you know that 1 acetyl-CoA produced how many NADH, friends? It has produced 3 NADH molecule. So 3 into 3. How many NADH molecule, friends? Total 9 NADH molecule, right? And friends, how many FADH2 molecules are produced, friends? All the students follow my words correctly. I am talking about 1 acetyl-CoA, friends. Only 1. So 1 into 2, we got 2 ATP. So now you calculate 9 ATP here, 2 ATP here, 1 ATP here. Total how many ATP, friends? 12 ATP. 12 ATP for how many acetyl-CoA molecule we have calculated? 12 ATP for only 1 acetyl-CoA. But we should always calculate for the 2. So friends, what you have to remember? 12 into 2, which is 24 molecules of ATP. So what will be the total now? 24 plus 6 plus 8. So total how many, friends? It is 38 molecules of ATP. Once again, every student follow me without confusion. When I use the word 38, every student has to keep it in your mind, friends. 38 will be the gross. Right. But if they ask you a question net, you should always consider 36 molecules of ATP. Then what happens to that 2 molecules of ATP? That 2 molecules of ATP we have used in the electron transport chain. So the total will be 36 only. Friends, now all of you has to understand the concept without confusion. What they are asking you in the exam. Remember my statements. They will ask the question like this. What is the total amount of, remember my words correctly, what is the total amount of ATP produced per 1 glucose molecule in the Krebs cycle? Remember my words correctly. What is the total amount of the ATP that is produced per 1 glucose molecule in the Krebs cycle? Then what will be the answer? Only 24 ATP molecule. See, that is where most of the students will get confused. And they will ask a question, how many number of ATP are produced for 1 glucose molecule in the glycolysis? If they asked you gross 8, if they asked you net 2. But whenever they have asked you a question very clearly stating that, remember the question, how many number of ATP are produced per 1 glucose molecule in the cellular respiration. What friends? In the cellular respiration. Then only you have to consider the glycolysis, then only you have to consider the pyruvate, then only you have to consider the Krebs cycle. See, 2 to 3 times the question only asked from the Krebs cycle, and most of the people will calculate for these 3 and they will choose the wrong answer. Sometimes they will ask the question only from the pyruvate decarboxylation, where the pyruvate convert into acetyl-CoA. Sometimes very specifically they ask for the more number of glycolysis cycles. Whenever they use a word oxidation of one glucose molecule in the cellular respiration, then only you people try to consider these 3, otherwise you have to calculate individually. And majorly remember friends, individually only. Okay. Okay. And one more unique statement. What is that one more unique statement? In this particular calculation, what I have done, I have calculated NADH all the time by considering 3 ATP, and I have considered 1 FADH all the time 2 ATP. Correct na? But friends, sometimes in the exam, instead of considering the 1 NADH as the 3 ATP, they will consider only 2.5 and they'll consider FADH2 as 1.5. So how you have to calculate depending on the answer they have given in the exam. Depending on answer that they have given in the exam, friends, you people has to follow that. Don't get confused here. Okay. Now, the most important one, which is the, see friends, I don't know whether you have seen the 4-marker question which is asked in this particular September 2022 paper. From this particular slide, we got the 4-marker question. Very straightforward question we got. We did not get the question from any calculation. We did not get the question from any, what we can say that any enzymes, any structure. We got a simple question. In the matching. Anyway, first you remember. In the glycolysis, how many regulatory enzymes we have studied? 3 regulatory enzymes. Friends, even in the Krebs cycle also, friends, there are 3 key regulatory enzymes are present. I really don't know what you do, friends. I want you to remember the 3 enzymes compulsory, and most importantly, you people has to remember their inhibitor compulsory, friends. If you forget their inhibitor, you can't answer the question at all. Citrate synthase enzyme. What's the function of a citrate synthase enzyme? You all people know that, which is converting this particular oxaloacetic acid plus acetyl-CoA, it is giving rise to the citrate. The enzyme is what, friends? Citrate synthase enzyme. I want you people to remember that if there is a presence of NADH, ATP and succinyl-CoA and citrate, if these 4 are present, friends, the citrate synthase enzyme will get completely inhibited. Then isocitrate dehydrogenase enzyme. If there is a presence of ADP, it is activated. If there is a presence of NADH and if there is a presence of ATP, they get inhibited. Don't confuse. ADP can acting as the positive activator or stimulator. NADH and ATP can act as a inhibitor. Then the alpha ketoglutarate dehydrogenase, same. It is also inhibited by ATP, succinyl-CoA, NADH. Here there is nothing to explain. You have to remember them compulsory, friends. Now all the students listen to me carefully. This is the slide which is the unique slide. Very simple, friends. Okay. What I am talking here, remember, I am using a line. Krebs cycle is, I will solve in the evening 7 PM PYQ classes. Focus. Krebs cycle is a source of what? Biosynthetic precursors. All the students follow me. You know very well, the acetyl-CoA combined with oxaloacetic acid, we are getting the citrate. Now you all people know that citrate is continuing the Calvin cycle. At the same time, citrate plays a crucial role in the production of fatty acids also, in the production of sterols also. 6 citrate, which is a 6 carbon. And what about the alpha ketoglutarate 5 carbon? Very important, friends. Alpha ketoglutarate, which is a 5 carbon, it is also continue the Calvin cycle. At the same time, friends, this can give rise to the glutamate, and it is also involved in the production of other amino acid, and it is also involved in the it is also acting as a precursor in the production of a purines. I want you to remember succinyl CoA, which is a 4 carbon, very important one, required for the porphyrin ring. I hope you know very well, right? What does the chlorophyll made up of, friends? Chlorophyll made up of the porphyrin and phytol chain. So actually, friends, that porphyrin ring is actually derived from the succinyl CoA only. And oxaloacetate even involved in the production of aspartate, involved in the production of purines as well as the pyrimidines. Okay, this is the area where they given a simple question which no student will read actually, right? Yes. Okay, friends, that's all for now. In the 11:30 a.m. class, we will finish the entire electron transport chain. We will discuss about the inhibitors. We will finish the oxidative phosphorylation, and we will also study the gluconeogenesis. By that, the carbohydrate metabolism will be completed. We will solve the PYQs. Right? Correct. Porphyrin head. Is it clear, everyone? Please let me know if anyone having any kind of a doubts. I will re-explain. And I am requesting all of you, please revise this PPT right now for 10-15 minutes and attend the 11:30 a.m. class. By that, you can remember the concept for easily. Clear, everyone? Any doubts, I will re-explain. Perfect. Friends, please revise. By that.